Hello and welcome back to your Chem 4 lecture. This week we're moving on into chapter 16 and chapter 17. 16 is about proteins and enzymes, which are a kind of protein. And then we'll move on to nucleic acids and your genetic information. So let's go ahead though. We're going to start by talking about proteins. Again, just like before, you're gonna have heard a lot of these words before that are used in your biochemical part of this class. But now we're going to put it in kind of new context, and we're going to show you the molecular structure that makes up proteins. So proteins are polymers, and what that means is they are basically made up of many small pieces called monomers. So those are the individual small pieces that add up together into a big chain. Again, that's called polymer. Now, the polymers that make up proteins can use any of 20 different monomers. These monomers are called amino acids for proteins. Now, proteins have a wide variety of functions. So there's structural proteins like cartilage, muscles, hair, and nails. Those are all parts of your body that need a particular structure to kind of give shape to the body. But there's also enzymatic proteins that function as a way to catalyze biological reactions. So in other words, it makes the reactions occur at body temperature and very specifically, and they're extremely important. We'll talk more about those in the next lecture. And then finally, you also have proteins like hemoglobin, myoglobin to transport oxygen in the blood. So these are just some of the functions of proteins. They're kind of the workhorse of the human body. They do a lot of these things. They're always at the active site of whatever is going on. So proteins are almost always involved in virtually any biological process. Okay, so here's our classification. So structural is basically just exactly like it sounds. It's just a structural component of the body. So you have collagen, that's a tendon and cartilage protein. That's what makes up, like your knee cartilage and stuff like that. Keratin is a protein that makes up hair. So, hair on its own is not living, right? It doesn't conduct nerve transmissions or anything. That's why you can cut it, or theoretically you could get it cut. And that's again just made out of protein. Then there is contractile protein, and these are kind of the functional parts of muscles. Muscles basically are made of these proteins that can contract and then, of course, relax, and that is what causes muscle movement. There's transport. So we mentioned this already about hemoglobin, transports oxygen. There's the lipoproteins that we talked about in the previous chapter, made out of proteins, and again, used to transport lipids in the body. There are storage proteins. So casein is an example of this storage protein. It's what stores protein in milk. And ferritin is something that stores iron in your spleen and liver. There are protein hormones as well. So we talked about hormones, steroids in the previous chapter. Proteins can also function as hormones. For example, insulin is a protein, and that is, of course, responsible for regulating blood glucose, like we talked about in the carbohydrates chapter. Human growth hormone, or growth hormone in general, is also a protein-based hormone. Next up, we have enzymes, which is a really broad class. Because they catalyze virtually every biochemical reaction that occurs in cells and in your body. Just a couple examples here. Sucrase is exactly what it sounds like. It cuts apart sucrose into its two monomers, and trypsin is the one that catalyzes the breakdown of proteins. Then finally, we also have the protection proteins, which include things like immunoglobulins. To stimulate immune responses. Okay, so let's start by talking about the monomers. It always makes sense in chemistry to start from the ground up. Now, as we're about to find out, we're going to hit the interface here where biology and chemistry meet. So chemistry is going to describe things in terms of the amino acids. We're going to build that up into protein descriptions, and that's where biology kind of takes over, is kind of understanding how the proteins interact with each other. But let's start with amino acids here. So amino acids, fortunately, have, there's 20 of them, but they fortunately have a very similar structure. So all of them always have an ammonium group here outlined in blue, and there's a carboxylate group that's connected by a carbon that we call the alpha carbon. Now, this central alpha carbon is going to be bonded to a hydrogen atom, and then it's going to be bonded to one unknown. So this unknown could be one of 20 different R groups, and this is what actually determines which amino acid it is. So again, this central or alpha carbon, that's going to be bonded to an R group. That R group is going to tell us which amino acid we actually have. So we have two different classifications for amino acids, and this is how we're going to start talking about them. We have nonpolar amino acids here, and then we have polar amino acids here. So valine is classified as a nonpolar amino acid. Its side chain is this propyl group here. It's an isopropyl group. And we can see that it's clearly nonpolar because it's completely composed of carbon-carbon and carbon-hydrogen bonds, which are nonpolar. So we again classify this as a nonpolar amino acid. Over here we have asparagine. Now asparagine is, the R group contains an amide. So we have here a carbonyl bonded to an NH2. Both the carbonyl and the NH2 are both going to be polar. They've got a polar carbon-oxygen bond, carbon-nitrogen, nitrogen-hydrogen bonds are all polar. So we would call this a polar. Now, the reason that we want to classify it this way is because nonpolar amino acids, like valine here, are going to be what we call hydrophobic. In other words, they don't want to interact with water. So they're going to be excluded from the outside of whatever place they happen to be in, unless they're like integral in the cell membrane, for example. So like if it's a blood protein, the hydrophobic amino acids are going to cluster in the middle where they're away from the water. Whereas if they're in a lipid, the valine is going to be happy being in like a non-polar environment. And it's the opposite for the polar amino acids here, including asparagine. Here, this side group, amide, is very happy to interact with water and other polar substances. So it would be exposed, for example, in the blood or in the cellular environment, but it would be interior if it's in, for example, a cell membrane. We'll talk more about this in a little bit. But here we go. Okay, so let's go ahead. We're going to classify them according to the three different ones here. Now, you don't need to memorize this necessarily. The only thing you do need to memorize is the name of each amino acid. And it's three-letter abbreviation. So, okay, and it's one-letter abbreviation. So here, let's zoom in because this is kind of hard to see. So we're going to start with the non-polar ones here. And the very simplest one, which is glycine. Now, glycine has two different, and all of these amino acids have two different abbreviations. There's a three-letter abbreviation and a one-letter abbreviation. And these are both going to be important. So this is really what you need to memorize is, given the name of an amino acid, what's its three-letter abbreviation and its one-letter abbreviation. So glycine is the simplest one because its R group is just a hydrogen. Okay, so glycine is GLY or G. Alanine is the second most simple one. That's just got a methyl group on it. And that's ALA, which is three-letter ALA, one-letter A. Valine we've seen here. Valine has this isopropyl group here. And again, it's abbreviated VAL or V. Leucine has a isobutyl group here. And this isobutyl group is, of course, nonpolar. It's all carbon-carbon and carbon-hydrogen bonds. Leucine's abbreviation is LEU or L. And then here, isoleucine has a sec-butyl group. So it's a 1, 2, 3, 4 carbon group. So a butyl group. And it's what we call sec-butyl formation. And isoleucine is abbreviated as ILE or I. So note here, the bottom of the structure is the shared amino acid structure. We have the ammonium, followed by the alpha carbon bonded to its single hydrogen, followed by the carboxylate. And then up here, that's where we have our R. Okay, so those are the methyl ones. Now down here, we're going to look at a couple more. We have first up phenylalanine. So phenylalanine has a one carbon chain attached to a benzene ring here. Then we have methionine. Methionine has kind of an interesting structure. And it also, because of this structure, it has a really interesting function that we'll talk about in protein synthesis, but we don't need to get there yet. Methionine, sorry, so phenylalanine is PHE or F, methionine is MET or M, and it's got this structure where you have two carbons in a chain, followed by a sulfur, so it's a thiol group or a thioether in this case, and then it's got a methyl group on the end of it. So that's your that's methionine. Again, that's going to be kind of the trickiest one to memorize, perhaps. Next up, we have proline. So proline is got its own special feature here, which is its side chain actually forms a ring with the nitrogen. So proline has this special scenario where it's got a 3-carbon chain that then folds back on itself, and the 3rd carbon bonds to the amino group, as shown here. So proline's the only one that does that, but that's the one that you need to know. And then finally, tryptophan. Tryptophan has this very complicated structure here, where the alpha carbon is bonded to a carbon, so CH2, and then it's bonded to this what we call an aromatic network, where we have a benzene ring with a neighboring 5-membered ring that contains a nitrogen, and that nitrogen is usually protonated as we draw. So that is tryptophan. Now, you might see this nitrogen ask, why isn't this polar? Well, because of this large aromatic group here, all these carbon, and carbon-hydrogen bonds are nonpolar. So this tryptophan is overwhelmingly nonpolar. Okay, let's get out. Go. Okay, so that's our first set of amino acids here. Those are the nonpolar ones. Okay, now let's look at the polar amino acids. So the simplest one is serine, which is abbreviated as SER or S. Here it is, these are the neutral ones. We have neutral and ionic next. Serine has a CH2 bonded to an OH, so it's almost like a methanol kind of stuck on there. Threonine here, very similar, but in addition to the hydroxyl group, there's also a methyl group on that carbon that's bonded to the alpha carbon. Remember the one in the blue, that carbon is the alpha carbon, and then it's bonded to the side chain up here. Threonine is abbreviated as THR or T. Tyrosine is abbreviated as TYR or Y, and it has this, one carbon bonded to a benzene ring, and then an OH on the other end. So it's a phenol-based amino acid. Next up, we have cysteine. Cysteine is abbreviated as CYS or C, and it has a 1-carbon chain bonded to a thiol, SH. Next up, asparagine. That's what we saw in the previous one. This is abbreviated as ASN or N, and it has an amide group here. So the amide group, again, is just a 1-carbon chain followed by an amide. Glutamine, closely related, still an amide, but it now has an additional carbon in between the alpha carbon and the side chain. So I should say in the side chain, between the alpha carbon and the amide. So asparagine is CH2, then an amide. Glutamine is CH2, then CH2, then an amide. And glutamine is abbreviated as GLN or Q. Okay, finally, we have these, we have 2 sets here. We have these carboxylate amino acids here. I want to point out here, I'm showing you these amino acids at physiological pH, which is roughly our blood pH here. And note that the carboxylic acids are deprotonated and the amine group is protonated. So this is the way, if you had a free amino acid in your blood, this is how it would look. So for the 2 acidic amino acids here, aspartate and glutamate, they look a lot like asparagine and glutamine. The only difference is, of course, now they have a carboxylate, or if they were neutral, they would have a carboxylic acid up here. So aspartate is ASP, or abbreviated as D, and glutamate is GLU, abbreviated as U. The only difference here being that aspartate has a single carbon connecting the alpha carbon and the carboxylate, and glutamate has 2 carbons connecting. Finally, we have our polar basic amino acids. And again, these are hydrophilic as well. And these are going to be protonated at physiological pH. So just in the same way that the carboxylic acids are deprotonated, the amines are protonated. So that starts with histidine, which is this structure here. Now you'll be given this structure in your notes on the test. So You don't need to memorize the structure, but again, you do need to memorize the name followed by its abbreviations here. So what histidine is, it's got a 5-membered ring with 2 nitrogens, and both of those nitrogens are protonated at physiological pH. And that's what that looks like here. Next up, we have lysine, which has 1, 2, 3, 4 carbons in its side chain. So a butyl chain that connects to an amine at the end of it. And again, at physiological pH, that's protonated as an ammonium. And then finally, arginine, which has this really interesting structure where we have 3-carbon chain as a side chain, bonded to an amine, so bonded to a nitrogen, that's then bonded to a carbon, and that carbon is bonded to 2 separate nitrogens. One nitrogen it's single bonded to, the other nitrogen it's double bonded to. And at physiological pH, the one that it's double bonded to is going to be protonated. So again, that's our arginine structure here. Oh, finishing up here, lysine is abbreviated LYS or K, and then arginine is abbreviated as ARG or R. Okay, so here we go. That's our structures here. We won't walk through that. Again, that takes a long time, but good to make sure to pay attention as you're going through. So remember, alpha carbon is going to be bonded to its 3 components. It's got a hydrogen bonded to it. It's got the ammonium and the carboxylate at a physiological pH. And then finally, a 4th component, which is the R group. Okay, so we already kind of covered this here. The only thing different between the amino acids is the R groups. Everything else stays the same. Okay, so let's go ahead and do a quick sample exercise. Identify each of the following amino acids as polar or non-polar. We have glycine and threonine. Based on their structures, tell me, are they polar or non-polar? Pause the video. When you come back, we're going to take a look at it. So pause, and let's take a look. So glycine, because it's just bonded to a hydrogen, that's a non-polar bond, so that's a non-polar amino acid. Threonine here contains an OH group, so that OH group is going to be polar, so threonine is a polar amino acid. All right, so this is going to take a little more practice, but you can go back in your notes here, and I want to see the structural drawing for each of the amino acids. And make sure to give me 3-letter and 1-letter abbreviations as well for valine and cysteine. So go ahead, pause the video, work it out, and when you come back, we'll take a look at the answer. So pause. Okay, let's take a look here. So valine has this isopropyl group. Cysteine is a thiol group connected to a carbon that's then bonded to our alpha carbon. So again, it's pretty easy once you draw the basic structure down here. You just have to put the side chain up above it. Okay, so those are our amino acids. Now we have to explore those first, but of course, that's just the beginning because we want to focus on proteins, right? Well, how do amino acids make up proteins? Well, the first thing we need to talk about is what's called a peptide bond, okay? And a peptide bond is a type of amide bond that forms between amino acids. Basically, one carboxylate group is going to form a bond to the ammonium group of the next amino acid. So this is what's called a peptide. So you're linking up these amino acids that become peptides, and this is a particular type of amide bond called a peptide bond. Okay, now if you take 2 amino acids and form a peptide bond, it forms a dipeptide. 3 would form a tripeptide, and 4 would be a tetrapeptide. And what this is basically is a chain of amino acids going from, think of it as going from left to right. And so, each amino acid can link up with up to 2 other amino acids, one on its carboxylate end and the other on its ammonium. So for example, let's take a look here. Let's imagine that we wanted to explore the structure of a peptide bond that forms between glycine and alanine. So what happens here is what you're going to do is you're going to say, okay, I've got glycine and alanine, and I want to form a peptide bond specifically between glycine and alanine. Well, what's going to happen is the O on the whichever one you have on the left is going to this is going to go away, and the carbonyl group is going to form a bond to the nitrogen over here. So we're going to get rid of water because we're going to lose the O from the carboxylate side of the left amino acid, and we're going to lose 2 H's from the nitrogen on the peptide or on the amino acid that's on the right side. So our new bond that forms is a carbon nitrogen bond here, and we lose a water molecule. So this is what we call a peptide bond. Again, it should just look like an amide. We've got a carbonyl bonded to a nitrogen, and that nitrogen carries on into the next one. Okay, so here's another kind of structural view of it. Again, you start with an ammonium. You're going to lose a water molecule here, this oxygen and two of these hydrogens, and then you form a peptide bond that just is that structure. Okay, so why don't you go ahead and give it a shot? Give me the dipeptide, draw the dipeptide that forms between SER and THR. So serine and threonine. Given the structures that you have here, go ahead and draw what the dipeptide is going to look like. Pause the video, take your time, walk it through, and then we'll take a look at the answer when we come back. So go ahead, pause. Okay, so this time what we're going to do is we're going to form a new bond from this carbon to this nitrogen. And we're going to lose this oxygen and two of these hydrogens, right? We're going to form a water molecule. So the dipeptide that forms is just going to have a peptide bond, an amide bond, between this carbonyl and this nitrogen. So now we have serine on the left and threonine on the right. Okay, so now how do you actually name these peptides? Well, there's a pretty simple naming scheme. So okay, so let's go ahead and first define N-terminus and C-terminus. So note that when we put these peptide bonds together, we're starting with an ammonium on the left side, or on the left side over here, and we're ending with a carboxylate on the right side. That's because these amino acids are not symmetrical, right? They have the ammonium group on one side and the carboxylate on the other side. So when we form these peptide linkages, we're clearly going to end up with a carboxylate on the right side, and we're going to start from an ammonium on the left side. So the side that starts with the ammonium is going to be called the N-terminus. The side that ends with the carboxylate, that's going to be the C-terminus. And that's the way that we can distinguish between these two ends. Now, to name the peptide, all we have to do is we take our names of our individual amino acids, and we cut the last syllable and replace it with YL. So alanine becomes alanyl, glycine becomes glycyl. And then the C-terminus, we just use its normal name. So serine just stays as serine. So to name this peptide that forms from alanine, glycine, and serine, we would call it alanyl glycyl serine. And that would be abbreviated as ALA, GLY, SER, or just AGS. Okay, so why don't you go ahead and give it a try. Again, it's not too complicated compared to the naming that we've done already this semester, but give me the 3-letter abbreviation and name for the following tetrapeptide. Remember, for the 3-letter abbreviation, all you have to do is put the 3-letter abbreviations, one following the next, separated by a hyphen. So go ahead and give it a try here for a tetrapeptide that forms between alanine, first on the N-terminus, then leucine, then cysteine, then methionine at the C-terminus. So pause the video. When you come back, we'll take a look at the answer. Pause. Okay, so the way that you would do this is, first off, ALA, LEU, CYS, MET. Those are the 3-letter abbreviations for our amino acids. And then follow it up by changing the names here. So alanine becomes alanyl, leucine becomes leucyl, cysteine becomes cysteanyl. I know that's confusing. And then methionine becomes just methionine because it's at the C-terminus. So alanyl, leucyl, cysteanyl, methionine. Okay, now here's another thing that you probably heard about amino acids before, which is that some of them are, quote, essential amino acids. So we have 20 amino acids, but only 11 of them can be synthesized by our body's biochemistry. The other 9 are stuff that we need to obtain from our diet. So this is the list of essential amino acids. That's histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Not having all 9 of these can lead to very specific syndromes that can be a problem with your health. So the kind of the most straightforward way possibly to get these is to have complete proteins in the form of eggs, milk, meat, or fish. However, not everyone, wants to eat all of this stuff. So you can also devise a diet that contains all 9 from plant sources, including grains, beans, and nuts. Those individually are deficient in 1 or more amino acids, essential amino acids, but usually when taken together, you get enough of each of them that you have a complete diet. Okay, now that kind of covers it there. I don't think I need to say anything more there, but let's go ahead and talk a little bit about proteins. What we've talked about so far, the building up of amino acids, this is slowly going to lead to protein structure. Now, in order to actually get a protein, by definition, you need 50 or more amino acids, all linked together, and they have to have some biological activity. That's the definition of a protein. Now, what we've talked about so far is what's called the primary structure. The primary structure, so this is number 1, this is the particular sequence of amino acids that is held together by peptide bonds in a given protein. That's what we call the primary structure. Now, so let's go ahead and take a look here. So there's a thyroid hormone, for example, that stimulates the release of thyroxine, and that is a tripeptide with the sequence of GLU-HIS-PRO. This is it here. So you could imagine having glutatine, histidine, and proline in any particular order. Like it could be pro, glu, his, or his, glu, pro. But the thing is, if you mixed it in any other order, it wouldn't have the same hormonal activity. Only in this particular order, glutamine, histidine, proline, is when you get the hormonal activity. And that's because clearly the structure going from left to right matters. So the primary sequence definitely matters in terms of proteins. If you were to mix this one with this one, you have a different shape of the protein, and therefore you have a different, or polypeptide, I should say in this case, the tripeptide, and you're going to have a different activity. Okay, how about insulin? Well, insulin is a relatively simple protein. There's 2 chains. There's a chain A with 21 amino acids and chain B with 30 amino acids. These are linked together with disulfide bonds. Remember, those are between cysteine residues. That's something that we talked about all the way back in the thiol chapter. Insulin was the first protein that had its primary structure determined. Again, that's because it's a relatively simple protein, but also a very important protein. And again, we have this primary structure. We have 2 polypeptide chains, and then they're linked together with these disulfide bonds. Okay, let's go ahead and take a look here. So we have a tripeptide that I'm showing you here. I have 3 questions for you about it. I want you to identify the amino acid that's at the N terminus, identify the amino acid at the C terminus, and then give me the name of the tripeptide. So go ahead, pause the video, find out what those names are, and come up with the name of the tripeptide. And when you come back, we'll take a look at the answer together. So pause. Okay, let's go ahead and take a look. So at the N terminus, remember that's the side that's got the ammonia here, and this should be H3 plus, by the way. Apologies for that. We have a benzene ring on a methyl group connected to the alpha carbon. That's phenylalanine. At the C terminus, that's the carboxylate end. We just have a methyl group on the alpha carbon. That's alanine. In between, we have cysteine. If we want to name it, that means it's phenylalanyl cysteanyl alanine, from, again, N terminus to C terminus. Okay, so let's go ahead and talk about how this links to health. So polypeptides, remember, this is going to be anything that's less than 50 amino acids is a polypeptide. We have 2 different types of natural painkillers called enkephalins and endorphins. Both of them are polypeptides, and they have particular receptors in the brain that basically shut off our pain response. So enkephalins are pentapeptides, and these are very small molecules. Because they have this pain numbing response, they are the smallest molecules that contain this opiate activity. Here, for example, is beta endorphin, which is 1, 2, 3, 4, 5, 6, 7, 8, 10, 11, 12, 13, 14, 15 amino acids long. So there's another couple of hormones that are polypeptides, oxytocin and vasopressin. Oxytocin is something that stimulates uterine contractions. So in order to kind of speed along labor or induce labor at the end of a pregnancy, you can give oxytocin. Vasopressin is an antidiuretic hormone. And basically that just adjusts the amount of water that your kidneys reabsorb. So diuretic means it makes you go pee more. Antidiuretic just means that it holds onto water a little bit longer. And interestingly, again, very similar structures. The only thing different is isoleucine substituted for phenylalanine from oxytocin to vasopressin, and leucine substituted for arginine as well. Okay, so why don't you go ahead and give me the names and three-letter abbreviations of the tripeptides that can form from 2 glycines and 1 alanine. So this is just kind of a thought exercise. Give yourself a little bit of time to think about, you don't have to draw the structures here. You just need to think, how could you organize these into 3 different tripeptides? Remember, structure matters. Okay, so pause the video. When you come back, we'll take a look at the answer. Okay, let's take a look at it. So if we have the 2 glycines first, we get glycyl, glycyl, alanine. Then we could have the alanine in the middle, that'd be glycyl, alanyl, glycine. Or we could have the alanine at the front, which would be alanyl, glycyl, glycine. And remember, these are very different. They could have very different activities in the body if they do have activity, because you have the difference between the N terminus and the C terminus. Okay, so that's the primary structure. But of course, that's not where it ends. In fact, there's a total of 4 types of structure of proteins. We talked about the primary structure, but now we need to talk about the secondary, tertiary, and quaternary structures that are going to form. Okay, so starting with secondary structure, there's 2 really important parts of secondary structure. A third, I think, that we'll talk about briefly. But the 2 main components of proteins here, or main secondary structures of proteins, are going to be starting from the alpha helix. So what happens in an alpha helix is hydrogen bonds form between the oxygen of the carbonyl of 1 amino acid, and then they form a hydrogen bond to the hydrogen bonded to the nitrogen of an amide bond in the next turn of the alpha helix. So here all the alpha carbons are kind of in the helix along with their nitrogens and their carbonyl neighbors. And so that's kind of forming the basis of the helix. And you have all of the R groups kind of sticking out from them there. And you have these individual hydrogen bonds forming. I think it's 5. Hold on, let me see. 1, 2, 3, 4. Yeah, so like 4.5 amino acids away. That's where you form this hydrogen bond. And that's what holds this helix together. Yeah, because you get this kind of long hydrogen chain. So this is where we go from basically, structures that look like this, that look like chemistry. Now in the biology simplification, we're going to have this kind of picture here. So again, we're kind of transitioning from our chemistry knowledge to now our biology knowledge or biochemistry knowledge. So that's one. The other one is a beta pleated sheet. And this beta pleated sheet basically occurs when hydrogen bonds again occur between the peptide backbones, where you have the hydrogen on one of the amides of one of the peptides is going to form a hydrogen bond to the oxygen from the carbonyl of the next one over. So now these beta pleated sheets can be, any number of distance away. There's a whole curve here. But it just depends on the particulars of each of each protein, how far away those are going to be. So again, this is what a beta pleated sheet looks like. Again, we have another type of hydrogen bond forming here, and then we have R groups pointing in both away from you and then up at you as you look down at the sheet. Okay. So now there's one other type here. There is also what's called a triple helix. This is not really as important. This is where you take 3 polypeptide chains and they can weave together in these ways here. hydrogen bonds are basically holding these chains together in the same way. Now, this toughness is particular in structural proteins. So think like collagen, anything else that's like a connective tissue, like tendons, skin uses these in cartilage because the connections are very strong. And yet collagen fibers are these triple helices. And again, it holds all three of these together through hydrogen bonding. Okay, so let's talk briefly about these secondary structures and a quick link to health. Alzheimer's disease is something that you may have heard of. It's a type of dementia that occurs in which a person experiences memory loss that increases, and then an inability to handle daily tasks. So I've had multiple members of my family have Alzheimer's or similar diseases, so maybe you have as well. What happens is basically in a normal brain, these beta amyloid proteins that are made up of about 42 amino acids have an alpha helical shape. In someone with Alzheimer's, these beta amyloid proteins seem to change shape. The normal alpha helix form goes away, and instead you have these beta pleated sheets, which are these like insoluble protein fragments, and they form plaques in the brain. So it seems like, again, this is not like, it's not totally established. We just have noticed that these plaques seem to correspond to a diagnosis of Alzheimer's disease, where you have a lot more of them if you have Alzheimer's disease. So this is the normal healthy structure that you get from these beta amyloid proteins. But again, in some brains, it just seems like they stop folding like this and they start folding like this. And these beta pleated sheets now form plaques that perhaps interfere with your cognitive function. So definitely the difference between alpha helix and beta sheet is significant. Okay, so now we've talked about the primary structure. That's the literal chain of amino acids. We've talked about the secondary structure. That's these hydrogen bonds that form either alpha helixes, beta sheets, or potentially triple helixes. Next up, we have tertiary structure. So the tertiary structure is how the overall three-dimensional shape is formed, basically by having interactions from one part of the protein chain to another. So you can have hydrogen bonds or cysteine linkages that form over long distances in the tertiary structure. In the secondary structure, those are all over relatively short distances. So here, these are always going to be determined by the cross links, which is basically attractions or repulsions potentially that are formed from the side chains of the amino acids. So polar side chains prefer other polar side chains. Non-polar side chains like other non-polar side chains. So like here, for example, if we look here at this, just an example of a tertiary structure here, you can have all sorts of different interactions between side chains. So here, if we have two phenylalanines next to each other, well, those phenylalanines are both nonpolar, so they're both going to be hydrophobic, and they're going to want to be next to each other. So we have this hydrophobic interaction that occurs here. Now here, what if we have a serine on one side and a serine on another side? Well, the hydroxyl groups of serine are going to attract each other through hydrogen bonding. So that's another example of, again, tertiary structure. Something called a salt bridge can form. That's where an ammonium side chain, so the amine side chain here, forms an ionic attraction to a carboxylate on another side chain. Hydrogen bonds can also form. Hydrophilic attractions can also occur. And then finally, disulfide linkages are also tertiary structure. So these are the 5 types of tertiary structure that can exist. The hydrophobic interactions, that's between any two nonpolar amino acids. Hydrophilic interactions, that's basically you have your external aqueous environment in your cell or blood, for example, and the R groups of polar amino acids, those are going to be attracted to it. Salt bridges are ionic bonds that form between, you have basic amino acids that are positively charged and acidic amino acids that are negatively charged. Those are going to form an ionic bond called a salt bridge. Hydrogen bonds, of course, we've talked about these before. That's where you have, again, the hydrogen from a polar R group interacting with an oxygen or a nitrogen of another polar R group. And then finally, disulfide bonds that are in particular bonds between linkages that have occurred between cysteine amino acids. So that's where cysteine has a thiol, that's SH, but you can form a disulfide bridge bonding the two by removing the hydrogens. Okay, so that was a lot of structure here as far as we're talking about secondary and tertiary so far. Why don't we go ahead and just kind of quiz ourselves a little bit on the different types here. So based on the statement that's given A through D, indicate the type of protein structure that is given to you there, whether that's a primary structure, an alpha helix structure, a beta pleated sheet, or a triple helix. So pause the video, go through this on your sample exercise sheet, and then we'll take a look at the answer when we come back. So pause. Okay, let's take a look here. So polypeptide chains that are held side by side by hydrogen bonds. Well, that sounds like a beta pleated sheet, where these chains are kind of stacked one on top of another in a sheet form. The sequence of amino acids in a polypeptide chain, that's the definition of a primary structure. A corkscrew shape that has hydrogen bonds between amino acids, that's an alpha helix. And then finally, 3 peptide chains woven like a rope, that is a triple helix. Okay, so now this is going to be a little trickier because you're going to have to go back and look at your amino acids. But given 2 amino acids here, select what type of tertiary interaction could occur between them. So like, for example, leucine and valine. Leucine and valine are both very nonpolar. So I would say that leucine and valine would have a hydrophobic interaction with each other. So pause the video. We'll take a look at the answers when we come back and come up with B through D here together, okay? All right, let's take a look. So 2 cysteines. Cysteine is the one that forms disulfide linkages, where the SH bonds are broken and you form a new sulfur-sulfur bond between 2 cysteines. Aspartic acid and lysine. Aspartic acid is a polar acidic, and lysine is a polar basic linkage, so or amino acid. So these would form a salt bridge or an ionic bond. And then finally, serine and threonine both contain an OH group, so you would have a hydrogen bonding group for both of them. Okay, so that leads us to the 4th type of structure, which is what's known as the quaternary structure. So the quaternary structure, now we take these individual proteins and we build up into a bigger unit. So this is how 2 or more polypeptides combine to form a single protein. So for example, hemoglobin has 4 different polypeptide chains, and they combine in a very specific arrangement to form hemoglobin. Now, the interactions are going to be the same as what we had from our tertiary structure, but now they're occurring between different polypeptides. So again, we take primary structure. Primary structure folds into secondary structure in a single polypeptide. That polypeptide then will fold further into a tertiary structure to give you, one particular polypeptide. And then that polypeptide can interact with other polypeptides to form the overall quaternary structure of proteins. Okay, so that's your 4 types, right? Primary, sequence of amino acids. Secondary, alpha helix or beta pleated sheet. Tertiary, long distance interactions within a polypeptide that we talked about, whether that's hydrogen bonding, salt bridge, disulfide bridge, hydrophobic interaction or hydrophilic interaction. And then quaternary, that's where you have those same interactions, but between different polypeptide chains, forming an overall larger protein structure. Okay, let's talk quickly about sickle cell anemia. So we're going to find out more about this in the next chapter, but there's all sorts of abnormalities that can occur in proteins based on the way that they're interpreted from DNA. So what happens is basically in a normal beta chain of hemoglobin, you have this look here, and then in the sickle cell anemia beta chain, you have a glutamic acid that's been turned into a valine. And this is a big change. Big change because glutamic acid is polar, acidic, and valine is nonpolar. So that nonpolar R group is going to be attracted to different regions than the glutamic acid. And so what happens is the red blood cells change. They no longer form a rounded shape to a crescent shape. And this means that they don't transport oxygen as well as their neighbors. So these sickle cell anemia hemoglobins that form can actually have real problems where these can clog small capillaries because they can stick together in like long distance, almost like the plaques that we were talking about at the brain before. And this can cause low oxygen levels in affected tissues. So you get these large fibers that can form, and again cause problems with your bloodstream. Okay, so we already talked about this. These are the 4 different types of structures of proteins. Don't need to dive into this too much, but let's go ahead in our sample exercises and identify which level of protein structure is represented by each of the following, A through D. So pause the video, take a look through your notes, and when you come back, we'll take a look at the answer. So pause. Okay, so first off, beta pleated sheet, that is by definition a secondary component of the structure of a protein. The order of amino acids, that is again the definition of the primary structure. A protein that has 2 or more peptide chains, that's quaternary structure. And then finally, a disulfide bond between R groups, that is tertiary structure, although that could also be involved in quaternary structure if I said between R groups on different peptide chains. Okay, let's talk briefly about the structure of proteins and how you can actually mess with the structure of proteins. And this brings up the idea of denaturation. Denaturation is the disruption of the bonds in the secondary, tertiary, and quaternary structures by a number of different processes. So the first one that does this is heat or other organic compounds can break apart hydrogen bonds and disrupt hydrophobic interactions. This is, for example, cooking. This is the difference between raw meat protein and cooked meat protein, is that it's been denatured by heat. Acids and bases can break apart the H bonds that form between polar R groups and disrupt ionic bonds, whether by a base reacting with an ammonium that was involved in a salt bridge, or a acid reacting with a carboxylate that was in a salt bridge. Heavy metal ions can interact with the disulfide linkages and basically take electrons from those to form solids. And then finally, agitation, for example, whipping, that basically stretches the peptide chains until they actually physically break. So that's, for example, whipping eggs. You see that the structure of the egg changes from when you start, cracking it to when it's been whipped. And that is a result of agitation. And again, you can't imagine the reverse process where you would take the whipped egg and turn it back into an egg, right? Because that is impossible. Same thing here for heat. Heat changes the structure here from, you see these very liquid proteins here to the denatured protein here. Okay, so here's your 5 different methods for it. You've got heating above 50 degrees Celsius, acids and bases, organic compounds, heavy metal ions, and agitation are all ways to denature proteins. And denaturing proteins means that you're going to stop their current function. Okay, here we go. Tannic acid is used to form a scab on a burn. An egg is hard boiled by placing it in boiling water. What is similar about these 2 events? Okay, I don't know why I gave this to you here. Both of them involve the denaturation of a protein. So the egg starts as like a soft protein, right? Ends up being hard boiled because it's heated. Tannic acid forms a scab on a burn by denaturing the proteins involved in the skin or the burned skin. Okay, that covers our first discussion of proteins. In the next lecture, we'll talk about enzymes. And from there, we'll move on into nucleic acids. Until then, I'll talk to you soon, and let me know in the comments if you have any questions. All right, bye-bye.